Hello, today I'm going to be talking to you all about the effects body size has on the rate of decomposition by looking at the changes in total body score and percentage mass loss. Although there is a complete lack of research surrounding body size and the role it plays on decomposition, there are a lot of known influencing factors which have been heavily researched. These factors include, but are not limited to, temperature, rainfall and humidity, along with other factors such as the pH of soil, moisture, air movement and pressure. When looking at cadaver decomposition in the UK, a suitable human analogue must be selected. This is due to ethical and legal requirements. Many research projects have used a number of animals such as mice, rats, rabbits and pigs, though pigs are generally the most favoured human analogues due to their likeness to human skin. Decomposition generally follows a sequence of events and can be broken down into 5 stages. These stages include the fresh stage, the bloat stage, active decay, advanced decay and skeletonization. The aim of this research was to establish if there is a relationship between the body size of a cadaver and the rate of decomposition. The objectives of this study are to identify the 5 stages of cadaver decomposition, to compare the rate of decomposition seen between the size populations, to determine accurate measurements of the human analogues, and to calculate and compare the percentage mass loss of each cadaver. Although there is a lack of research surrounding body size and the role it plays on decomposition, 2 comparable studies were found that tested the theory that body size does have an effect on cadaver decomposition. These studies were conducted over 2 decades apart and demonstrate non-comparable results, even though they adopted comparable methodologies. In 1991, Howardicum and Goff observed an accelerated rate of decomposition in the larger cadavers during the earlier stages of decomposition, though their observations demonstrated a less conspicuous change in the rate of decomposition during the later stages. However, although Sutherland et al. Distinguished the relationship linking the initial carcass mass and the rate of decomposition, when measured in accumulative degree days, the results suggested that smaller cadavers reach skeletonization twice as fast as the larger cadavers. Numerous decomposition studies, although challenging different hypotheses, adopted comparable methods. After comparing many methodologies, it became apparent that the methods implemented by Sutherland et al. In 2013 would be a suitable basis for this research. The length of study was amended due to the utilisation of considerably smaller cadavers. This was in accordance with the study conducted by Stokes et al. In 2009. It was recorded that decomposition takes less time in smaller animals and therefore a shorter study period could be seen as a reasonable adjustment. The choice to use smaller cadavers was mostly due to the cost, as in the UK pigs are sold based on weight. It must also be noted that there was a lack of space available for this study, and thus being another reason to use cadavers with smaller body mass. The gap between the cadavers was scaled down in accordance with the difference in cadaver size, as well as the space available during this study. Less frequent data collection was also taken due to the temperatures in the UK being far lower than those in South Africa, where the study by Sutherland et al. Took place. This was decided in accordance with Gennard, who stated that the lower temperatures inhibit decomposition. And 3D scanning was chosen as it is praised for its non-destructive nature. This is what the experimental setup looked like at the facility. In order to look at the effects body size has on decomposition, 2 varying size groups were utilised. There were 2 groups of 3 pigs, 3 smaller and 3 larger cadavers. The smaller cadavers weighed on average 6.89 kilograms. Both cadavers are in scale of each other. Can you take a guess at the size of the larger cadaver? The larger cadaver weighed 14.58 kilograms on average. Numerous data collection methods were applied during the study, including total body score, point cloud data, mass loss, rainfall, and temperature. The total body scoring system used in this study was that of Magessi et al. 2005. Other methods, such as Myberg et al. 2017, were explored, but due to the lack of scientific scrutiny and experimentation, they weren't used. Unfortunately, the 3D scanner available was not up to the job. This meant that accurate measurements of volume loss and surface area loss could not be obtained. Therefore, the mass loss was recorded to ensure enough data had been collected for statistical analysis. Since the 3D scanning aspect of the experimentation failed miserably, mass data collection was recorded at weekly intervals. Here's me struggling to weigh the cadavers during the bloat stage, without them just sliding off the tarpaulin. This is one of the smaller cadavers, so you can imagine the struggle with cadavers that weighed twice as much. My technique was to use a digital fishing scale to record the mass of the cadavers, by hooking each corner of the tarpaulin onto the hook of the digital scales. The error margin for this technique was less than 5%. Here is one of the larger cadavers trying to escape one of the cages I'd made. Okay, so what about the results? During the earlier stages of the study, the larger cadavers decomposed at an accelerated rate. However, it was the smaller cadavers that reached skeletonization first. Inter-observer reliability was reasonably consistent during the study. However, please note no statistical testing has been completed at this stage. In week 1, a green tinge on the larger cadaver was observed. This is referred to as marbling. This happens during autolysis, which is essentially the breakdown of red blood cells. The larger cadaver is at maximum bloat, however the smaller cadaver is in the earlier stages of bloat. In week 2, the smaller cadaver is now entering the bloat stage. However, the larger cadaver is now post-bloat and the abdomen has started to concave. In week 3, insect colonization suggests that the smaller cadaver is in the active decay stage. This stage is when cadavers lose most of their biomass, which is due to insect colonization. The lack of entomology, on the other, shows that it is nearing the end of the active decay stage or in early advanced decay stage. At the end of week 3, the smaller cadavers overtake the larger cadavers, and much more decay towards the rear of the smaller cadaver was observed. Here you can see that during the first 3 weeks the larger cadavers decomposed at a slightly accelerated rate, though from week 3 until the end of the study it was the smaller cadavers that decomposed at a slightly accelerated rate. The sequence, however, is very consistent between the two parties. At week 7, both parties were scored lower than they were the previous week. This correlates with heavy rainfall during data collection. As you can see, the mass loss between the cadavers was pretty consistent throughout the study. However, during the first 3 weeks, it was the larger cadavers that lost the biggest amount of biomass. This is likely due to the amount of bacteria present, which ultimately kickstarts the process of decay. Where the cadaver seems to gain weight during the study, this can be explained by heavy rainfall. When looking at the graphs side by side, you can see that they are very closely linked. This is interesting as both graphs show a strong relationship between mass loss and the total body score. As the total body score increases, the biomass decreases. Every week, 3 independent scorers scored each cadaver. This was done to determine whether the inter-rater reliability was present. As you can see in the graph, the scores awarded by each scorer were very similar from week to week. This showed that there was some inter-rater reliability present during the study. However, please do note that no statistical analysis has yet been completed. Okay, so what about the significance of results? A better understanding of decomposition and its processes, a wider understanding of the impacts and whether they influence the decomposition sequence, knowledge that is likely to benefit forensic practitioners, especially when investigating juvenile deaths. It was also noted that rainfall severely distorts total body score, another contributing factor that should be considered when estimating post-mortem interval. The studies showed various flaws in existing taphonomic techniques. The most significant limitation was the weather conditions. There was rainfall each week, with the exception of week 3. The temperatures were considerably higher during the earlier stages of the experiment and continued to fall from week 3 of the study. The heavy rainfall present during the study also made applying total body score far more difficult, as it was hard to tell if the cadavers were wet from decompositional fluids or the rain. It also made it difficult to determine if the remains were dry during the more advanced stages of decay, because of the heavy rainfall present. The space available from the start was a huge hurdle, as a bigger gap between the cadavers would have ensured that insect colonization was controlled more efficiently. This is because the cadavers were only placed 50 centimeters apart, and larvae travel away from the carcass in order to pupate. Another limitation was the cost of the cadavers. Had the money and the space been available, it would have been beneficial to source cadavers with a bigger difference in body size. Although used in a wide range of taphonomic studies, the scale devised by Magessi et al. In 2005 is outdated and problematic. It was found during the study that it was very difficult to apply a single score to the cadavers. An analytical balance would have also been more fitting for this project, as they give far more accurate results. However, I'm not aware they make ones big enough to weigh animal carcasses. Further work would be beneficial in this field, as knowing how body size affects cadaver decomposition could be extremely important when investigating the death of a juvenile. The use of cadavers with a bigger size difference or a wider variety of size groups. Using specialized pods to control temperatures, humidity and rainfall would be beneficial. Exploring the methodology with human cadavers in human taphonomic research facilities in the States or Amsterdam or even in Australia could be something to look into. The development of a more accurate and reliable total body scoring system is also hugely beneficial in this area of study. So, to conclude, the body size of a cadaver has an impact on the rate of decomposition. The scoring system used may not be the most reliable technique. There is plenty of scope for further research in this field of study, especially on the development of a more reliable and user-friendly system for measuring cadaver decomposition. Thank you for listening. Any questions would be now welcome. Thank you.